Given that I'm interested in the brain, I need to use brain tissue to see if prenatal stress causes any changes to the brain cells. So what I did was I first generated prenatally stressed rats. So rats that experienced stress in the womb, and then I also had another set that didn't. They were fine. They were completely left alone. Happy ratty life. I then took these two groups and then split them each into individual groups, and one set got a week of chronic stress, or as I call it, a week of hell, and the other set didn't. So in each group I had prenatal stress, non-prenatal stress, prenatal stress, non-prenatal stress. Doesn't matter right now. I then put them through the ringer with a bunch of behavioral tests to see how anxious they were, because we know that prenatal stress actually tends to increase anxiety behavior in male rats, not in female rats. Interesting. I then took the rat's brains so that I could examine what actual changes were happening on the cellular physiological level rather than just behavior. This will give me an idea of pathways to target, or specific genes or proteins that might be of interest that might be affected by prenatal stress. And if I can find out what they are, then I can find out what causes prenatal stress, and then I can find out how to prevent it or to treat it. So at the end of last year, I spent a lot of time cutting brains. I had a lot of brains to cut. When I say cutting brains, I need to cut my brains into millimeter thin slices so that I can then use them for the next step of the experiment. I can't just use a whole chunk of brain for what I'm doing. So I use a machine called a cryostat, which is basically a glorified meat slicer like what you'd find at the butcher. It's also a freezer. It's a very multifunctional machine. So basically, I take a brain and I mount it on a block or a chuck with something called OCT, which is this white gloopy substance that freezes really quickly and keeps the brain in place. So now it's stuck on this chuck. I then put the chuck in the machine, close the guide plate, and use a lever on the side of the cryostat to move the brain up and down. So with each rotation, the brain moves up and down once. Because it's moving against a blade, that generates a teeny tiny 15 millimeter thin slice. So now I have this little slice of brain, and I take a room temperature microscope slide that's also coated with other chemicals that makes the brain tissue stick to it easier. And because of the change in temperature, it just sticks on. So I take that, stick it on. There you go. I've got a brain slice now. So I start at the top of the brain, the way it's mounted, so basically here, and cut my way all the way through to the back, because I want a bunch of different sections, because I'm interested in several different brain regions that are involved in stress and anxiety. Most of the sections that I cut and put on these microscope slides go back in a freezer box and go back in the freezer at minus 80 degrees Celsius until I actually need them for the experiment. However, every 4 slides, I take something called a marker slide, and the marker slide lets me see where I am in the brain. So that one gets dipped in a fixative. It's a formaldehyde mixture, like the kind of thing you would use for embalming. Give it a quick wash, dip it in a blue dye. You can look at it under the microscope, because before it was completely see-through. You can't see anything on that. Now, all the cells are stained blue, so I can actually see where I am in the brain. That means that later, I can go back and say, look, I just want to do an experiment using hippocampal sections. So sections of just the hippocampus. So I can go to my marker slides and see which 4 slides correspond to that region. Then go and pick out slides 50 to 54 rather than having to guess, oh, it was probably around 50 to 54, because 1, that's a waste of time, and 2, it's a waste of money. And this is just an overall easier way of doing this. After I finished cutting the brains, I could use these sections to do the next set of experiments, which was an in situ hybridization. And in that experiment, I can look at different changes in proteins or genes, more specifically, to determine if prenatal stress has an effect. And that will be in the next video.